All right, it's 12:30, so we'll get started. Small group today, but we'll make it work. So last time on Monday we started talking about Chapter 7. We talked a lot about hemoglobin and myoglobin and how their different structure helps them have pretty different functions. And today we're going to continue on that theme of protein function being affected by structure, and we're going to talk about muscle contraction. So let's start with those muscle contraction questions. So just to get everyone started, we'll just do some matching of different terms relating to muscle contraction. So everybody take maybe 5 minutes and start matching some of these terms to each other, and then we'll go over it together. All right, so that should be enough time to have gone through these few. So let's start out with number 1. What is the contractile unit of a myofibril? Anybody? Yeah, D. Yeah, it's a sarcomere. Oops, forgot to turn my pen on. Great. So what is the direct source of energy for a muscle contraction? Yeah, ATP, exactly. And so you might have noticed there was also another choice on here, glucose. So glucose is a nutrient in our food supply that we also use to derive energy from. The keyword in this question is direct. So ATP is directly used to fuel muscle contraction. Glucose is an indirect source because we use glucose in our bodies to make ATP, but the direct source here is ATP. And all right, so what is a long multinucleated cell? Yeah. Yeah, a muscle fiber. So this might be a little confusing, which is why it's on here. So a muscle fiber is a cell. It has multiple nuclei inside. There are myofibrils that make up the muscle fiber or the cell, among other things. But the unit that's a cell in a muscle is called a muscle fiber. So make sure you get that definition or distinction. All right, so what is the model we use for how structural changes will shorten the sarcomere? Yeah. Yeah, it's the sliding filament model. And we call it that because there are filaments that slide past each other that help to shorten the sarcomere. So it's named just like for what it does. Okay, so now let's start thinking in more depth, in more detail. So now we're going to look at a number of different proteins that make up the different, that each have a different role that they play in muscle contraction. So everybody, again, take a few minutes and match each of these descriptions to the correct protein. All right, it looks like most people are done thinking about this one. So let's start with number 1 again. So what is the primary protein component of thin filaments? Yeah, actin, exactly. What about thick filaments? Yeah, myosin, yep. And what is the protein that coils around and along the thin filaments? Yeah, tropomyosin, exactly. And what protein is present periodically along the thin filaments? Yeah, troponin. so that's kind of a description of where they are, and now we'll talk more about what they do. So what protein triggers muscle contraction after it binds to calcium? Yeah. Troponin again, yeah. So troponin helps move out of the way when it binds calcium, and that helps tropomyosin move out of the way, which makes space on the actin. So what now is the motor protein that undergoes the actual conformational change that makes the muscle contract? Yeah. So tropomyosin, it does move out of the way, but it doesn't take on a conformational change. It just shifts away from the binding sites, right? What is the protein that's actually changing its shape? Yeah. Myoglobin. So myoglobin we talked about on Monday, right? Myoglobin, yes, myosin. No, you're good. That's why it's on there, right? To make you think. So, okay, so let's tackle both of those. So myosin is the part, what makes up the thick filament that's in the sarcomere. The head on the myosin is what undergoes a conformational change to cause the sarcomere to shorten, and we'll talk about that in a little bit. Myoglobin is a protein that we talked about on Monday. I'll get to in just a second. And that's the protein that is there to capture oxygen after hemoglobin brings it from the lungs through the bloodstream to myoglobin, which is there in the muscles. And myoglobin helps to act as a store and a delivery of oxygen to the muscle cells so that they can make more ATP, which powers the muscle contraction. So they both start with myo. It's a little bit confusing, so make sure you know one's myoglobin, one's myosin. They do very different things. So yes, what was your question? So as far as I'm aware, the thick filament is composed almost entirely, if not entirely, of myosin. It's just many different myosin molecules that are all twisted together. And so that's why we call it the component of a thick filament, because it's not just like one myosin molecule, it's many, many myosin molecules. Any other questions about these proteins? Okay. So now let's think about the structure of myosin. So, yeah, so does anybody want to tell me about the subunits of myosin and if they're the same or different? Yeah. So it is, okay, so you're thinking of part of the myosin molecule, but not the whole myosin molecule. So myosin, as you can sort of see as we look further in the question, it has heavy chains and light chains. So I think you're thinking of the two heavy chains, right, that twist around each other, and they're the same, which is true. But myosin as a whole is made up of how many proteins? Yeah. So it is it is not. So it has, anybody else have any thoughts about this? Yeah. So there are, yeah, exactly. So there's 2 heavy chains and 4 light chains. So what does that mean for the total number of subunits we have? What's 2 plus 4, anybody? Yeah, so what kind of a mer is it? It's a hexamer, yeah. And are they all the same, or is at least one of them different? Yeah, it's hetero, exactly. So 2 are heavy and 2 are light. They're not all the same, so that makes it hetero. There's 6 total, so that gives us a hexamer. 2 of them are heavy, 4 of them are light. And heavy and light just refers to the size. So the heavy chains are larger, they're very long. The light chains are pretty small. And so let's move on to the last part of this question. So the domain of the protein where the catalysis occurs is called what part of the protein? Yeah, the head, yeah. The myosin heads undergo a conformational change, and this ultimately helps to shorten the sarcomere. Any questions about structure of myosin? Okay. Okay. Yes. Yeah. Actin is made of many repeating small subunits. Yes. Okay. Yep. Yeah. Any other questions about the structure? Okay, so now let's start thinking about the steps of muscle contraction. So you have 10 different steps, I think, here. So you can start to put them in order. Just keep in mind that this is step 1. Calcium binds to troponin. And then you have 10 steps that happen after that. So it's not just getting the order correct. Because remember, muscle contraction is a cycle, right? So you have to think about both the order that things happen in the cycle and where we're entering the cycle. So we have step number 1. Then we're going to have 2 through 11 underneath. So everybody take a few minutes. You can work with a neighbor on this one. It's a little bit trickier, but that's, yeah, we'll come back together in about 10 minutes and we'll talk about it. Yeah. Or it could be I think at that point, like if you lost the whole limb, I don't know that the loss of troponin would necessarily be reflected as like coming back into your bloodstream. Yeah. Just as a All right, so let's start talking about these steps in muscle contraction. So who knows where we start after calcium binds to troponin? I think the first one is fairly obvious. Yeah, maybe. To me, yeah. Yeah, exactly. So I'm going to label this step 2 because we started with step 1. So we know that we talked about before how after calcium binds to troponin, that's what lets the tropomyosin move away from the binding sites on actin. I would say the step after this is more tricky because now you have to know where in the cycle you're jumping in. So where did we pause while we were waiting for calcium, and therefore where are we going to pick back up? So does anybody know what would happen next? Yeah, G. That is a good guess, but that is not where we start. But good thinking. Any other ideas? Yeah, F. It is also not F. It's close, but not quite. So it's okay if everybody's stumped. It's not super apparent, I guess. So let's start, or let's look at the actual steps in muscle contraction. So this can, hopefully looking at this will give you a sort of better idea of what's going on. So this gives you an idea of the order of the cycle, of the order that all the things happen. But looking at this, we still have to think about what calcium is doing and where we would pause when there's no calcium. So we just talked about how when you add calcium, that binds to troponin, which allows tropomyosin to move away from the binding sites on actin. So that means without calcium, the binding sites on actin are being hidden, right, by tropomyosin. So you know that if there's no calcium around, myosin cannot bind at all to actin. It's waiting somewhere and is not bound to actin because the binding sites on actin are hidden by the tropomyosin. I'm getting some confused looks, some nods. So if actin, here's our actin polymer, right? So I'll just start by telling you it's here. This is where we'd have to wait. So essentially what you're looking for is the step between where myosin is not stuck to actin and where it's going to stick onto actin, right? So at this point, actin can wait here forever, right? It's not attached, myosin can wait there. It's not bound to actin. So if tropomyosin is hiding the binding sites on actin, it has to wait there. But if you try to go to this step, at this step, the myosin head is binding to actin. So for this step to happen, you need calcium because then the binding sites on actin are no longer hidden. Yeah. Sure. Yeah. So, yeah, I'll just start by saying in this picture of this cycle, it's assuming that there's just calcium there, and that's why the cycle can keep going around. So once you add calcium, once calcium gets released from the sarcoplasmic reticulum, and it can bind to troponin, the cycle can go around and it can keep going around. But then at some point, the calcium all gets sequestered away back in the sarcoplasmic reticulum again, and that's what tells the muscle to stop contracting. And so that's sort of that's where it had to start and stop. So the reason why calcium isn't drawn here is it's because You're just sort of assuming there's calcium there. But you can imagine, so I'm going to go back to the starred step. So you can imagine if calcium leaves and tropomyosin is going to bind back on, there's going to be tropomyosin all along the actin here. And so myosin can't stick to the actin because the binding sites are being hidden. That could also be happening here, right? Because myosin is also not attached here. So if calcium left the cell, you could have tropomyosin hiding the binding sites on the actin. But you wouldn't get stuck at that step because you can proceed to this step without myosin binding, right? Myosin doesn't bind to actin until you get to this step. So calcium is required to move on to that step. That's shown on the bottom here because the myosin head is binding to the actin during that step. Is that clear for everybody? Yeah.
That means muscles are compressed, they're kind of already bound to ATP, just waiting for the calcium to.
Exactly. Yeah. So there's always muscle cells, as long as they have enough nutrients or oxygen being supplied to them, they're going to be making ATP. So they're ready to go when they get a signal to start contracting. And so even when your muscles are at rest, they're usually at rest here. And as you pointed out, ATP is bound over there. So keeping that in mind, let's go back to our order of events, right? So what is the next step? Yeah. D, exactly. Myosin binds weakly to actin. So that's what we show here. It says right here, myosin binds weakly to actin. So the next thing that happens, I mean, you can just go along with this diagram, right? So what's the next thing that happens? Yes, it is J, where the phosphate is released. After the phosphate is released, that causes a change where the myosin binds more strongly to actin. So that's F. And at that point, very quickly, we get what? Once you get that strong binding, what immediately happens? Yeah. A. Yeah, the power stroke. Hopefully I'm getting these numbers right. Okay, so after the power stroke, now we've reached here, right? We released phosphate. We bound very strongly with this transient intermediate. So we had strong binding, and we very quickly did that power stroke. Now the myosin head moved relative to the actin. At that point, now what's happening? Yeah. It would be H. Yeah, so myosin is now going to release the ADP. And now ADP is released, which we show here. And so now we have our myosin head is now bound to nothing. There's no ATP or ADP bound, but it's still bound to actin. So what's going to happen next? So before it unbinds from the actin, one more thing has to happen. Yeah, so now ATP is going to occupy this empty site. So ATP is going to bind, which is G, and I think we're at 8. And then once ATP binds, now that allows the release from actin, right? So that's 9. Okay. And then, so now we're here, we released actin, and then what happens now? We released actin, we have ATP bound, and then, according to this diagram, the ATP gets hydrolyzed. Yeah. And when the ATP gets hydrolyzed, the myosin head cocks. So that hydrolysis interaction releases some energy that allows the myosin head to once again change its conformation. So myosin hydrolyzes ATP, that's 10, and then the last thing happens is the head changes its conformation. And now, assuming that there's still calcium in the cell, the cycle could keep going. Or if the signal stopped and the calcium is once again all sequestered away in the sarcoplasmic reticulum, this is where the cycle would have to pause until there's another signal that gives calcium. So you don't need a burst of calcium every single time the cycle goes around, but you do need calcium present, because once the calcium goes away, you are hiding, right, these sites on the actin, because the tropomyosin will move back into place, and then you get stuck right here where the star is. Yeah.
Probably,
Exactly, yeah. So once myosin is bound, the tropomyosin is not going to like shove it off. So as long as the myosin head is stuck on the actin, the cycle can complete. It's only at the point where the myosin voluntarily releases from the actin that the tropomyosin could like slide back into place. Yeah. I think probably. I mean, if you tear a muscle, presumably depends how deeply you tear it and where you tear it. But ultimately, to contract a muscle, you need a signal from your brain that tells the calcium to get released in your muscle. So if something happens that interrupts that signaling, that allows your brain to tell the muscle cell to release the calcium, then again, you'd get stuck in that same place. But if something else happens, like if that particular sarcomere just gets like chopped in half or something, like if that's where you tore it, then it would probably just stop at that wherever it was in the cycle. Probably depends on what happened. Yeah. Yeah. Okay. Yeah. Did I misnumber something?
I just
Yeah.
don't understand why the myosin binding ATP we have at the end when that in the diagram is like before the week
Yeah.
binding.
Okay. Yeah. So that's just because it's a cycle, right? So it depends where you want to start the cycle. It tells you what's at the beginning or the end. So like in the book, the way it explains it, I think it starts right here in the middle, and I think that's how I went over it like in the recording. But like in real life, what's happening really to make a muscle contract is it needs that signal from calcium. So in this question, we asked if you start with your calcium signal, that's going to change where in that cycle is the beginning or the end. Really, there isn't a beginning or an end. It's a cycle. So it's not like ATP is binding at the end. It's just that like when we paused here, like at our star, ATP had already been bound from the last cycle. So it's not like it just appeared out of nowhere. It's there were cycles before and there will be cycles later.
So calcium would not be present at the top before step one, like the very top where the arrow is pointing up.
Like over here where the picture is of the sarcomere.
Yeah.
Yeah.
Wouldn't be calcium yet there?
So you can do that step if there is no calcium, provided that there was calcium at some point before that allowed the myosin head to bind. So basically, like let me get a different color. So like in this part of the diagram, myosin is bound to actin on the right. And then in these two steps shown, or that's the left, sorry, I don't know right and left. On the right, myosin does not bind to actin. So you need calcium to expose the places on actin for myosin to stick. So you can't cross this blue line in this direction unless there's calcium present. So if to get over here where you were talking about, you needed calcium at some point, because like at some point you were stuck here, like from a cycle. And then if calcium, if there's a signal and calcium goes away, maybe you started the cycle, there was calcium. Maybe at some point, 10 cycles later, you're over here in the cycle, the calcium leaves. You'll keep going around, but then you'll get stuck back at where the star is. Does that kind of make sense?
I think so.
Okay.
Like if I started with the calcium binding, could I still then have the next step be like the AT? Like could I start with calcium and then go around starting at the blue? But I have to have the step you have right after that. Does it not matter?
So it does matter because you're assuming you're going from if you're going from no calcium to yes calcium, you'd have to start here because this is where you would have gotten stuck after when there was no calcium. So to start again, so like when calcium was removed from the cell, the cycle could continue to that point. So that's where you would be stuck. Okay. Sorry. Is everybody okay? Are we good? Yeah.
One of the videos like calcium like
Yeah.
So what part of this cycle is calcium? Or is there no specific?
There's no specific time in the cycle for it to leave. That's more regulated by like action potentials and nerves and stuff. Like this is just like the physical sliding filament model. So like this cycle can go around when there's calcium there and it will get stuck when there's no more calcium. There's no particular time when calcium needs to get like removed or added. Like it can get added wherever you are, but if there is no calcium, you'll just get stuck here. That's the main thing about it. Yeah. Yeah, so if there's no calcium telling your muscle to do anything, all of your myosin heads will be stuck right there. They'll have an ATP in them that's hydrolyzed, and the head will be cocked, and it will want to bind to actin, but it can't until the calcium comes and allows it. Yeah. Okay. So yeah, that goes back to just the idea of I don't know if I have that picture in these slides, but so like the way that actin and tropomyosin and troponin are arranged. Let me see if I can pull that up real quick. I think I have the other slides open. Maybe not. Okay. So here's a picture, right? So in the picture, you have your actin is yellow, your tropomyosin is blue. Or sorry, your actin is red, I think. Sorry. And then the troponin are like the little green things that are sort of holding the tropomyosin in place on the actin. So once calcium binds to troponin, the next thing that's going to happen is that tropomyosin can move out of the way. So that's not shown in that diagram with this cycle of muscle contraction because that's related more to calcium binding. It's not related to it allows the filaments to slide past each other. It's sort of like the on switch in a way, but it's not part of like the actual myosin actin interaction. It's just preventing that interaction. So if you're taking away the thing that prevents the interaction, then the cycle can begin. Does that kind of help?
Yes. How do you know? How do you just know that tropomyosin moves out of the way? Like, how can you just tell this?
I mean, not necessarily from the image. The image is more to give you an idea of where everything is. I mean, there's yeah, I don't know how to explain it necessarily. So it's like it's you can't see it, I guess, in the image. You can sort of think of it as troponin is like holding the tropomyosin in place. So if calcium moves the troponin away, the tropomyosin will no longer stay in place. Okay, cool. Yeah. 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 The blue. Yeah, so troponin is a trimer, so I think it's more than just the green. That's why I got confused, because there's a lot of colors. Tropomyosin is definitely blue. Actin is definitely red. I think the other ones might all be troponin. I'm not 100% sure on that. I can double check that thing for you. Okay. Any other questions about muscle contraction? All right. So that, if we're done with muscles, that will bring us now to immune response. So let's start by taking a few minutes. Start switching your gears to the immune system. So start defining all of these terms, and then we'll go over them together in a few minutes. At that moment,
He bound the it bind it. Oh wait.
wants to stick with. All right, so who can tell me what a pathogen is? Yeah. Yeah, so that's a good start for a definition. It's a foreign object. It's usually alive, so we can usually call it an organism. I guess it can also be a virus, which isn't alive necessarily, but for simplicity, I'll call it an organism. And it's true that it enters your body, but an important thing about a pathogen versus another type of microorganism that might invade your body. So not all, yeah. Yes, thank you. That's what I was getting at. So yeah, it's an organism that causes disease. So we have lots of microorganisms in our bodies all the time that do not make us sick, like all the bacteria and viruses and fungi in our microbiome, for example. They're just fine. They're supposed to be there, so they're not pathogens. Microorganisms are pathogens if they cause disease. Okay, so what is an antigen? Yeah. Yeah, excellent. So I'll call it a molecule. It is often a protein. And where is it usually found on the pathogen? Yeah, on the surface. Cool. So what is an antibody? Go for it.
It's a specialized cell that
Okay, you're close. You're on the way there. So is it a cell or is it another type of component? Yeah, it's a protein. Yeah. So antibody is a protein. It's made by cells, so it's one step removed. So what kind of cells make antibodies? Yeah, B cells. Yep. So it's a protein made by B cells, and it and it recognizes and binds an antigen. And you're right that it helps our body kill them, but it doesn't do that directly. So what is IgG? Yeah. Yeah. And what type of thing is that? Yeah. Yeah, exactly. So IgG is short for immunoglobulin G, and it is a type of antibody. So all antibodies are immunoglobulins. There are different ones that are all named with different letters that have slightly different functions. Yeah. I'm sorry, what? I'm sorry, one more time. Yeah, so immunoglobulin G are just the most abundant one that you find in your bloodstream. It's it causes most of the immune responses that we see when we think of like attacking and killing pathogens and things like that. There are other ones, you might have heard of IgE, that usually mediates allergic reactions. There's also IgA that's specifically found on certain surfaces in your body, like in your mucosal surfaces. You don't need to know all these, but there's IgM, which sticks to other types of antigens. So IgG pretty much only sticks to protein antigens. If you have antigens made of other stuff, they'll probably stick to IgM. But yeah, there's a variety. All right, so what are B cells? Yeah. Yeah, so they are white blood cells. That produce antibodies. So what does it mean to be autoimmune or have an autoimmune response? Yeah. So that would just be an immune response. So the key word here is auto. So what does auto mean? Self. So what would it mean to have an autoimmune response? Yeah, it's when your immune system attacks yourself. Exactly right. It can be mediated by antibodies that recognize yourself. It isn't always, so I'll just say often. So if you have antibodies that attack endogenous or your own cells, that would be an autoimmune response. That's an example of an autoimmune response. All right, any more questions about these terms for the immune system? I see some thinking. Okay. All right, so let's move on to the next set of questions. So these are true or false questions. Let's just go with it. So does number one, each IgG antibody has 4 antigen did you have a question? Yes. Endogenous. Sorry. Yeah. Yep. So does each IgG antibody have 4 antigen binding sites? Yeah. False. How many does it have? It has 2. All right, number 2. When a person is exposed to a new antigen, their B cells design and produce new antibodies towards the pathogen. Yeah. So, is it true? Does everybody think it's true? No. Okay. Yeah. Yeah. Exactly. So, these B cells, or your body makes B cells, has already made a lot of B cells, right? So your body makes all kinds of different, basically randomly generated B cells that can recognize almost anything. There are like millions and millions of different B cells in your body that can recognize millions and millions of different things. And so when a pathogen enters your body, one of the B cells that you've already made will already have an antibody that already happens to recognize that pathogen just through essentially random recombination. Yeah. So yeah, the problem is the time it takes to ramp up that immune response. So when you're just making lots of B cells, you have lots and lots of B cells, but each individual B cell makes a different antibody. So that's not very efficient if you have a whole bunch of like viruses that got into your body, because you need a whole lot of B cells that make that one particular antibody that recognizes the pathogen. So part of your immune response is basically like presenting the pathogen that enters your body to all of your different B cells, or a large number of all of your B cells. And then when it hits a B cell that sticks, then that particular B cell gets a signal to proliferate, to divide a lot of times. So that way you make a lot of, and that particular B cell, that original one, will clone itself, right? It'll make that particular antibody. And so you would get that one particular clone would expand, right? You'd get a lot of copies of that one B cell that makes that one antibody that recognizes the pathogen. But it takes time for all of those B cells to divide, and it takes time to find that one B cell that matches. So what a vaccine does is it exposes your body to a pathogen and lets that response go. It takes about a week or two. And that way, if you're exposed to the pathogen for real, you've already created a memory to that pathogen. But it's not that you created an antibody from scratch.
B cells are part of the acquired, right?
Adaptive, yeah. Yeah. Mm-hmm. Yeah, you are constantly making new ones, but it's not that when you are exposed to a pathogen, you make a special one just for that pathogen.
Creates that independent state in your body, not that it really happened. And that's why I get confused. We're literally the same part of the microbiome.
Yeah, oh, that's funny. Yeah.
I thought.
Yeah, so what happens is B cells aren't, when you just make B cells like in your bone marrow, they're not being made for any particular purpose or to recognize any particular thing. You just make a whole bunch of them, and it occurs it's basically random. Like you have all this recombination of all these different pieces that get mixed and matched. And so, and then you have these B cells. They're patrolling your bloodstream, they're patrolling your lymph tissue. And so when a pathogen comes into your body, particularly in the lymph nodes, you'll have this opportunity for all the B cells that you already have in your body to like look at the pathogen and be like, do I recognize this? Do I not? And if it does recognize it, then you have the growth of that one particular B cell.
So it's like a love where you might have a response, but you might also not have a response, and like you have like you randomly produce responses to like let's say like random codes, right? Mm-hmm. So you might have produced a code that was like that, and you might also not have. That's why you kind of need the person from that team to make sure they have that response to that one.
So yes and no. So there were a couple of different points I wanted to make. So I'm trying to think of how to like simplify it. So like yeah, no, it's not off topic. It's important to understand. It's just that, so I want to make it clear that like when you are producing B cells, the number of combinations that you make is so great. Like it is very likely that you have a B cell somewhere in your body that will recognize basically anything. So it's not like you have to be lucky that you made a particular B cell that recognizes the pathogen. You probably have one somewhere because there's so many different kinds of B cells. That's the first thing. And then the second thing was on a particular pathogen, like a virus, there's a whole lot of stuff that we're going to get to a vocab word about this in a little bit, but there are lots and lots of proteins with lots and lots of different parts of all those proteins on like the surface of a pathogen. So it's very likely that you have many B cells that will recognize different parts of that pathogen. So not only do you have a B cell that recognizes it, you probably have a few or several. And so you can expand out all of those different B cells. But you want to be able to expand out those particular ones so that you're ready for when you are infected. That's what vaccines are for, so that you're like prepared and you already made a lot of copies of those B cells that already recognize it. Does that kind of help?
Yeah.
Okay. Is everybody good? Okay. All right. So let's move on to number 3. So polyclonal antibodies. This is kind of what I was talking about just now. So polyclonal antibodies bind to multiple epitopes on the same antigen. Yeah, true. Yeah. So we sort of just talked about this, but so one antigen can have multiple different epitopes. So like if this is your antigen, it's a protein that's on the surface of something, you might have one region over here that this antibody can bind to. You might have another region on the other side that some other antibody can bind to. And I can keep trying to pick new colors, but you kind of get the idea. So that you can have one antigen that has multiple different sites on it that different antibodies can recognize. And each recognition place is called an epitope. Does that make sense for everyone? Epitopes versus antigens. Yeah. Yeah, so polyclonal, so that comes from when we were talking about how many B cells recognize your antigen. So one original B cell will make exactly one particular antibody. And if that B cell proliferates and divides, all of its progeny are going to make the same exact antibody. And that would be a monoclonal antibody because they all arose from one original B cell. Polyclonal antibodies would be like what you would see in your body and like when you get infected with the pathogen. So you'll have multiple B cells in your body that will recognize different epitopes on that antigen. So like in this example, you had 3. And one made the red one recognized the yellow one recognized the green one. And so in this weird color scheme. So that means you had 3 original B cells that each made different antibodies that all recognized the same antigen, just in different locations. If you had those 3 B cells, each of these antibodies is different, right? And they are from different, what you'd call B cell clones. So like it's polyclonal because you had multiple different original B cells so all of the antigens are similar because they recognize the same antigen, but they're all different because they recognize different epitopes. So a monoclonal antibody would only recognize one epitope on the antigen because it arises from one original B cell. Polyclonal antibodies arise from more than one original B cell. How did I do? Okay. Yeah. I would say most of them do. Yeah. It depends where you got them from or how you made them. So like if I got infected with SARS-CoV-2 and I made an immune response against it and I made a bunch of antibodies against SARS-CoV-2 and you took my blood and you looked at my antibodies, that would be a polyclonal response. I would have had a whole bunch of B cells that made a whole bunch of different kinds of antibodies that recognize different parts of SARS-CoV-2 or different parts of the spike protein. And that's a polyclonal response because I'm recognizing lots of different things using lots of different B cells. A lot of times, what's that? Right. It would be monoclonal, but that wouldn't happen because antibodies are so specific. So there are Okay, so there are special engineered antibodies that can recognize more than one thing, but that's not what we're talking about here. Like bispecific antibodies recognize different things, but don't worry about those. But like a lot of times in the lab, we want to use monoclonal antibodies that only recognize one particular epitope. So like when we talked about ELISAs a while ago, so like if you think about, this is like your surface of your ELISA, you have your antibody that binds to the plate, and then you have your antigen that sticks to that antibody. So that one antibody will recognize one epitope of your antigen. Then you would have a different monoclonal antibody that's your secondary antibody, because that's going to have to recognize an epitope that's on the other side of the antigen. So that makes sense. You'd have, in this case, you'd have two different monoclonal antibodies that each stick to different parts of your antigen. But they each stick to this, one sticks to the bottom and one sticks to the top, or whatever. So like different purposes for different things. Yeah. Yeah, you wouldn't. Yeah, like it would be really hard to pick them out from like a clinical sample or something. You'd have to do work to like purify the antibodies. Yep. Cool. Any other questions about this? Cool. All right. So let's talk more about the structure of IgG. So that's kind of why we're here, right? We're talking about protein structure and function. So, yeah, so I guess take a minute, maybe look at your IgG notes, and then we'll talk about the secondary, tertiary, and quaternary structure of IgG. Yeah. So like can one piece of an antigen combine multiple antibodies?
Sorry. I think the body can bind multiple antigens.
Yes.
Okay.
Yeah.
At the same time. Okay.
Yeah. So combine two. All right, so let's talk about the structure of IgG. So does somebody want to start us off with the quaternary structure? Like thinking about subunits. How many do we have? Are they the same or are they different? Yeah. So yeah, you are definitely on the right track. So antibodies have two heavy chains, and then they also have two light chains. That's really not a great picture, but yeah, so how many pieces is that? Yes, it is a heterotetramer. Excellent. It has two heavy chains and two light chains, and those chains are also linked together by disulfide bonds, which I did not draw in this weird little picture. But you can imagine they're kind of all over the place. I don't remember exactly where they are, but they hold it together. Okay, so what about the tertiary structure? Yeah. So yeah, so there are disulfide bonds that stabilize the tertiary structure. So remember that domains are part of the tertiary structure, right? So what kind of domains do we see in an IgG antibody? You just said it like a minute ago. It's like the different pieces. Yeah, there's constant regions and there's variable regions, and each of those are different domains. So like in my dinky little picture, we can kind of outline where those are. So most of the structure is pretty constant. Like this is constant. The bottom part of this is constant. There's also variable parts, and those are just the parts on the end. Sorry, there's a much better picture coming up in a couple of slides. Like, please don't draw this. But then the variable regions are right on the ends, the part that actually recognize the antigen, right? So, No, I just made random circles. Yeah, there are a few different constant domains. It's more recurrent in the real picture. You'll get it. Okay, and does anybody remember the name of the particular like folded piece that we get in there? It's okay if you don't. Okay, so each domain that we see on an IgG is made of an immunoglobulin fold, which is made of a beta sandwich. So it's a beta sheet that's like folded on itself to make a little sandwich. And those particular beta sandwiches are stabilized by disulfide bonds. So yeah, and that's all part of the tertiary structure because they're part of the 3D folding of aspects of the secondary structure, which we will now get to. So that hopefully gave a little bit of a clue as to what are the big components of the secondary structure. Yeah, beta sheets. Yeah, so beta sheets, and do you remember if they're parallel or anti-parallel? They're anti-parallel, yeah. And so those are what make up the parts of the beta sandwich. And what else do we see in terms of secondary structure, especially in the variable parts? Yeah. Exactly, yeah, the hypervariable loops. So those are the two big things we see. We got anti-parallel beta sheets. Those are in those beta sandwiches, which are part of the immunoglobulin folds that we see in the constant regions. And then there are, in the variable domain, hypervariable loops that, like their name says, they vary from antibody to antibody or from clone to clone and help your antibodies each recognize a different thing. All right, any questions about the levels of structure? All right, so let's everybody take a few minutes. Here's the better picture that I was just talking about. So everybody take a few minutes, and we just talked about all these different regions and pieces of IgG. So go ahead and label the structure, and then we'll go over it together in a few minutes. Okay, so let's get started labeling this diagram. So anybody can tell me any piece of it and where it goes. Let's see how that works. Yeah. Yeah, so we have light chains. What else do we got? We have the heavy chains. Yeah. And where are those? Exactly. Yeah. So here are the heavy chains. All right. What else do we have? Yeah. Sorry? The hinge. Yeah, so in the middle here, we didn't talk too much about that, but yeah, that's the hinge region. Yeah. Yeah, so the hypervariable, or the variable region. That's where we see our hypervariable loops. Yeah. Yeah, so the yellow is constant region. So I'm just going to point at the yellow part and say constant domains. Yeah. Okay. What else do we have here? Yeah. Yeah, so we have antigen binding sites. So we have one over here. We have some more over here. What other kinds of secondary and tertiary structure can we see here? Yeah. Yeah, there's a whole bunch of disulfide bonds and they stabilize a lot of stuff. I'll just point at a few of them, but we have a whole lot of disulfide bonds. There's more that I didn't point to, but you get the idea. And yeah, we're missing one other big thing, I would say. Yeah, exactly. The beta sandwiches, which are also called the immunoglobulin folds. What other color do I have? It's purple. Here is a beta sandwich or an immuno. All right. Yeah. Yes. Yep. So yeah, so like the ones in pink are the ones that are going to change. And so you can see even in this picture, they look like the 6 that are shown in yellow. That's 6, right? Or no, there's 8 that are shown in yellow. I can't count. They all look exactly the same structure and shape-wise. And then the ones that are in pink, they kind of started out as a beta sandwich, and then, but because they have these super hypervariable loops on the ends, they have like extra loopiness to them. So they form like a slightly different shape, if that kind of makes sense. Yeah. Yep. Yeah. Any other questions? Yeah.
Wait, the people that are still considering the same change?
Mm, I would say no, but I would have to look that up to double check. It's possible you could maybe like consider it as like kind of a beta sandwich. But yeah, I'll look that up for sure. Sorry. Yeah. Yeah. So there are multiple constant domains. So like this one, like this heavy chain that we're looking at has one variable domain and 3 constant domains. Is that, I don't know what color I made that, but okay, if that makes sense. Okay, yeah. All right, any more questions about IgG? Cool. So let's start talking about antibodies. So how have antibodies been used in the detection, prevention, and or treatment of COVID-19? So anybody can shout out if they think of anything. Any ideas? Yeah.
Or actually
Mhm.
have vaccines generating more antibodies for the virus.
Yeah, exactly. So how about in terms of detection or treatment? Yeah. Yeah, excellent. So for treatment, there are several different companies that made monoclonal antibodies. These monoclonal antibodies recognize one particular epitope on the spike protein. Different companies might make different ones, or even I think some companies made ones that were what they called a cocktail of monoclonals, where they had a couple of different monoclonal antibodies in the same mixture, so they called that a cocktail. And so something else that was also used more towards the beginning of the pandemic that also involved antibodies was convalescent plasma. So yeah, so convalescent plasma is the plasma or the liquid component of blood from a person who has recovered from the disease. So in the liquid part of their blood, that's where their antibodies would be. So if you were sick and you recovered, your blood will have lots of antibodies against the pathogen. And the plasma, or the serum, I guess, from your blood could be used to then to take advantage of your antibodies to help someone else use your antibodies to fight off the infection. Yeah, so what about detection? Does anybody know about any tests for COVID-19 that might involve antibodies? Yeah. So PCR actually is looking for RNA, so it's looking for SARS-CoV-2's genome. It's not using antibodies. Yeah. What other types of tests can you do? Has anyone ever done an at-home test, like a rapid test? Does anybody know how those work? Probably not. So, yeah, they use antibodies, exactly. So the in a very simple way, the rapid tests, they're called rapid antigen tests. They're looking for SARS-CoV-2 antigen, right? They're looking for bits of spike protein or bits of nucleocapsid or whatever protein. And the way they do that is in like the liquid part that they give you of that test, there are antibodies that recognize pieces of SARS-CoV-2. There's multiple ones. And in a very simple way, if those antibodies can find bits and pieces of the virus in your saliva or whatever, they will bind to a piece on that strip, also through antibody binding, and cause the line to turn color. So there's like a color change reaction that involves antibodies. I won't go into it. You don't need to know the details, but there are antibodies involved. If anyone's interested, we can talk more about it later. So, and then one other way antibodies are used in detection. This was more frequently used like early on in the pandemic, but you might know of someone, or you yourself might have had an antibody test to see if you were infected with SARS-CoV-2. Maybe you've heard of that, maybe not. So particularly early on in the pandemic, people were very concerned if they had been exposed or been sick with it. And so you can go get your blood drawn. They'll look in your blood and see if you have made antibodies against the virus. And so the main difference between looking for antigens and looking for antibodies, antigens will tell you if you currently have the pathogen in your body, right? The antigen is part of the virus. So if you have virus that's in there, that's hanging out, you'll have virus antigen in your saliva. An antibody test that's looking for antibodies that you made yourself, that's looking for if you had an infection in the past. It doesn't tell you whether or not you have an infection right now. All you're looking at is, did your body make antibodies against the virus? And that could have happened anytime. It doesn't have to be right now. All right. So any questions about different ways we use antibodies? Okay. So we'll just spend the last few minutes talking about the different COVID-19 vaccines in a pretty relatively simple way, just to sort of get an idea of how vaccines help you make an immune response that helps you recognize the virus. Yeah.
When it comes to like antibody testing, they're like in labs. Are they looking at like your white blood cells or vaccine?
So they're looking for just antibodies. So your B cells are a type of white blood cells that make antibody, and when they're activated, they'll start to secrete them. So they'll release the antibodies into your bloodstream. They're no longer attached to the cell. They're just floating around like in the liquid part of your blood. So yeah, they're just in the liquid part of your blood. So they're, along with the red blood cells and the white blood cells, they're antibodies, and there are a lot of other protein components that make up your blood. Yeah. Cool. Okay, so yeah, let's talk about COVID-19 vaccines. So we're starting off with the mRNA vaccines. These are probably, if you've been vaccinated, you probably got one of these. These were like the most, I would say, popular ones. There are two companies that made the ones that are approved in the US, Pfizer in partnership with BioNTech, and also Moderna, which are written over here. So the way that these work is that, so as we know, the SARS-CoV-2 virus has a protein on the outside, the spike protein. And so in a lab, these companies, made mRNA that has the code for the spike protein. So they make that mRNA artificially. There's some minor changes to it so that it is not going to cause problems in your body. But ultimately, it makes a protein that looks very similar to the spike protein. That mRNA is encapsulated, and it's not drawn here, but it's encapsulated in a lipid nanoparticle. And that helps to protect the mRNA, like in your body, because RNA by itself is very fragile. It's very sensitive. And there's lots of things in your body that if they see RNA just floating around, they'll just like chop it up. So to protect that from happening, they're enclosed in a lipid nanoparticle. Then you have this RNA vaccine. And then this is sort of showing that lipid nanoparticle, which is kind of over here. It's a really bad color to use. Over here, this lipid nanoparticle is able to fuse with your cells, and that's going to let the mRNA that's part of the vaccine enter your cells. And as we know, mRNA has instructions for how to make a protein. So the ribosomes in your cells are going to take those mRNA instructions and make a spike protein that can be now on the surface of your own cell. And your immune system will have B cells, at least one of those B cells makes an antibody that recognizes the spike protein. And that B cell then is given instructions to make a whole lot more antibodies and also to divide a lot. And ultimately, and a lot of other things happen. This is very simplified, but ultimately you end up with a memory that you've seen this spike protein before, and the next time you see it, you can respond to it very quickly and kill off the pathogen. So that's, in a nutshell, how the mRNA vaccines work. Another type of the COVID, you have a question? Yeah. So the actual mRNA part of the vaccine doesn't stick around very long in your body. Your body uses it to make protein, and then the mRNA eventually gets broken down and recycled. And the spike proteins eventually all get eaten up by components of your immune system because they're not part of your body. And so what remains after that vaccine is memory in the form of antibodies that are patrolling around in your bloodstream. And also there are special types of immune cells that are called memory cells that live a long time. And so when you get a booster, that kind of reactivates particularly those memory cells, and it tells them like, hey, wake up and make some more antibodies because we just saw this pathogen again, we're in trouble. And another difference, especially with the COVID-19 boosters, is the actual mRNA sequence is slightly different in some of the more recent boosters to reflect some of the changes that have happened in the virus. So to reflect the different variants, they have slightly different sequences on their spike protein. So the mRNA sequence, that's the actual vaccine component, is reflective of the more recent, those mutations. Cool. Okay, so that brings us to the viral vector vaccines. So viral vector vaccines in this country, they were made by Janssen or Johnson & Johnson. There's also AstraZeneca also made one that I believe was never approved here in the US. So these are similar but different. So in this case, the DNA that codes for the spike protein they take that in a lab and they put it into the code for a virus. And so the code to make the spike protein, the instructions are in another virus that's been made harmless, and that is the actual vaccine. So then here we have our harmless virus that has the instructions for making a spike protein, again, enters your cell. And then once again, the cells in your body use the instructions that are in that harmless virus to then make the spike protein. And then you get the same sequence of your immune system recognizing the spike protein and making that memory to it. The main difference is the vector. So either way, you're giving your cells instructions for making spike protein. But in the case of the viral vector, the instructions are made from DNA instead of RNA, like they are in the mRNA vaccines. And just trying to get through this before the class is over. So the last main type of vaccine we're going to talk about for COVID-19 is the protein subunit vaccine. So this one that's approved in the US is made by Novavax. So This one's a little bit different. So instead of giving your body instructions to make the spike protein, the company makes the spike protein in a lab. And they do that using, they actually use insect cells to do this. So they put the instructions to make the spike protein in a type of virus that infects insect cells. And then those insect cells, they're actually from moths. So the moth cells, once they're infected with the virus, they are given instructions to make a lot of spike protein. And so then ultimately you have these moth cells in a lab making all the spike protein for you. That spike protein is then purified, so you have just the pure protein. And that protein, along with what's called an adjuvant, is what the actual vaccine is made of. So you have purified spike protein that was made in a lab, and added to that is a substance called an adjuvant that helps just to generally boost your immune response. And then that is what goes into your body. And so once again, you have spike proteins now in your body. You have your own body's immune system has antibodies that recognize the spike protein. You make your immune memory. So those are the three main types of COVID-19 vaccines approved here in the US. In other countries, there are other types. There are a lot of different types of vaccines around, but we're just going to focus on those three for the purposes of this class. Just really quickly, if anyone's interested in more about this stuff, Dr. Middleton had pointed out there's some interesting stuff on this podcast, and there's also this article that she found that is about the persistence of antibodies after infection or vaccination. And then in the rest of the slides, there is more information about actin filaments and muscle growth and exercise if anyone's interested. We're not going to be tested on this, or you're not going to be tested on this, but if you're interested, there's more resources in here. And so with that, I just want to remind you all, quiz 5 is due tonight, and have a great weekend. We'll see you Monday.